Yeah. Okay. What are you Googling?
Stuff.
Oh, okay. Oh, we're rolling, by the way.
Oh, are we? Okay.
Yeah, we're totally rolling. Like that Fred Durst song.
I don't know who that is.
He was the guy that was all about science projects.
No.
Yeah. Get with the program.
I just realized I have BB cream on this half of my face, but not on this half.
Baby cream?
BB cream.
Oh, BB cream.
Yeah, it's like foundation.
Oh.
But lighter.
Sounds like a rapper. BB cream.
BB cream.
Yeah. Or it's like it sounds like a cartoon character that's like a play off of BB King.
Mm-hmm.
But he's like made out of like Cool Whip or something. And his name is BB Cream. And he's like the smoothest guitar player. All right, you're good.
Okay.
I don't know, but this is pretty cool.
I know, right?
There you go.
I made that.
I like it.
Okay.
You can start off by saying, hello, friends.
Hey guys! Today we're gonna pull DNA out of a strawberry. How's that sound? I did this once in high school, and it wasn't with a strawberry, it was with a piece of some sort of cow anatomy. I don't remember what part, but it was gross. It was gross. Strawberries are way better. Okay, so we're gonna need water, salt, dish soap, very cold alcohol. This is just like 90% isopropyl, like rubbing alcohol. Just stick it in the freezer for a couple hours. And strawberries, obviously. Need some measuring spoons. I'm gonna use these for like scraping stuff out of my measuring cups. A little stirring stick maybe, or if you're a nerd like me and you have your own lab glassware, a glass rod, and a plastic bag. You're also gonna need something to strain your liquid through, so you can use either, if all you have is paper towel, that should work. Cells are very, very tiny and they should be able to slip right through a paper towel. You could also use cheesecloth, or like even a piece of regular cloth, or a mesh strainer. You could use that too. If you use a mesh strainer, you might want to get like a spoon or something to kind of squish the strawberry mush down in, so it doesn't like just sit there. So let's get started. I'm so excited. Okay, so the first thing we're gonna do is, what was the first step? Okay, so we need 90 milliliters of water. So this is my 100 milliliter beaker. A beaker is shaped like this. It's not shaped like that. This is an Erlenmeyer flask. A beaker is straight up and down on both sides. It has a little spout. So we need 90 milliliters of that. Measure carefully. I can feel you judging me over there for having my own lab glass.
I kind of am. No, it's more like just me being envious.
I can do science anytime I want.
Oh.
Then we're going to need 10 milliliters of soap. It's going to be just to that. Yeah, because that's like milliliters. Fun fact, a milliliter and a cc, or a cubic centimeter, are the same amount. That is the same volume of fluid. Okay, so we have our 10 milliliters of soap, 90 milliliters of water, and we're going to mix those two together. So I'm going to kind of scrape that in here. Stir it up gently so it doesn't overflow.
Gently now, gently.
Okay, and then we need a 1/4 teaspoon of salt. The salt what does the salt do? I don't remember. So the salt, DNA is hydrophilic, which means it wants to be close to water. The salt helps to make it more hydrophobic, so it will separate from the rest of our solution.
So you say it's a little clingy?
It's a little clingy, maybe, yeah.
Ain't no clingy.
Quarter teaspoon of salt. Mix that up. Come on, dissolve. I don't have the patience for this. It's taking too long. I want results now.
Breathe in, breathe out.
Okay, there we go.
You need to set an example.
Okay, so the next thing we're going to do is get our plastic bag. And then get a strawberry. So let's pick... Let's see, a good one that doesn't look like I want to eat it later. Oh, this one's kind of ugly. Let's use that one.
Hey, come on.
It's giving its life for a good cause. In the pursuit of science. The pursuit of learning. Okay, so strawberry goes in there. This mixture, which is it has a name, our extraction buffer. So the soap is going to dissolve the cell membranes. A cell is like a very tiny part of a living organism. It's like the tiniest part you can get without being down to like molecules, basically. There are smaller parts of cells that are inside cells, but we're not going to discuss those right now because they're exhausting. So salt pushes water away, and oh yeah, so the soap helps keep the DNA from degrading too. Preserves it a little. So this is going to go in here. We're going to squeeze out as much air as we can. Very carefully so we don't get soapy salt water all over our cool Pikachu shirt. Okay. And then we're going to smash it. Fail. This is the fun part.
You're so violent.
You're just jealous.
Yeah,
You're just jealous you don't get to squish a strawberry.
I'm a little jelly that I can't make some jelly.
Oh, that's so gross. It's so gross. It feels gross. Oh, God.
Maybe. May I?
Carefully, don't burst that.
Oh!
It feels so icky, doesn't it?
Never again. Never!
I think you should use a second strawberry.
It's up to you.
Okay. Let's use a second strawberry. And that was kind of a small strawberry. Maybe a big one. Nope. Squishing it will help to break the cell walls and free the DNA that's inside of the cell walls, or inside of the cells, rather. Now, DNA is basically your body's instructions for you. It tells your hair what color to be, what color for your eyes to be. It tells your body how tall to grow, what color your skin is going to be, what shape your earlobes are. It even tells your body to grow a third tendon or not behind these two in the front here. That is a thing that is genetic.
Sounds a bit bossy.
Genetic. Genetic means it's in your DNA. It's something that can be passed from parent to child, or from grandparent to grandchild, if it's something that skips a generation. Gosh, dang it. Come here. Dang you. He's a strawberry. And DNA inside of a cell is a very, very tightly coiled double helix. Now this is a double helix. It's two strands that spiral, and these spiral together, and between them are our base pairs, which is like, it's sort of like computer code, but for living things. Base pairs are, they're made up of the nucleotides adenine, thymine, guanine, and cytosine, and I believe adenine and thymine bond to each other, and then cytosine and guanine also bond to each other, but cytosine and adenine aren't gonna bond, and guanine and thymine aren't gonna bond. So it's always AT or CG. So let's continue squishing. Almost done, I think. Oh, that's still very cold. Now the cold of the alcohol also helps to keep the DNA from degrading while we're trying to get it out of the strawberry. Okay. I think that's good. What do you think? Looks good to me. That's the right color.
Yeah.
It looks disgusting, doesn't it?
I mean, it looks like something from an 80s horror movie. So
I love things that are disgusting. Science is often kind of gross.
Yeah.
That's what I love about it. Okay. So, oh gosh. Next thing we're gonna do is strain our extraction buffer with our squished up strawberry in it.
That sounds like a cool device.
Hmm?
Like something.
Extraction buffer?
The extraction buffer.
Okay.
Like a mad scientist would use that.
Just a little extra squishing, just in case. Okay. So I carefully open it. It smells nice. It smells way better than it looks. I'm gonna pour some in. And we're gonna watch that pretty pink fluid pool at the bottom there. Now this liquid, that pink liquid, is our extraction buffer, but it's full of cells, broken cells, and free-floating DNA strands. So remember how do you remember how I told you that a DNA strand is a very tightly coiled double helix? Those double helixes are coiled so tightly that they're microscopic. We can't see them without a microscope. But if you stretch out a single DNA strand as far as it'll go, it'll be like 9 or 10 feet long. Just one DNA strand. And you have millions or billions or trillions of strands of DNA in your entire body. I don't even know how many cells are in a human body. It's a lot. It's a lot. Okay, I think we can put a little more in here. Cool. Oh, God. It's so icky.
Now it just looks like medicine.
Gross medicine. It's a pretty color though, right?
Pepto-Bismol, that's what it looks like.
It's such a pretty color. I got new glasses. I feel like a superhero. Oh! Oops. Oh no! Guess what?
Oh no.
I dropped it. It's fine. That's fine. It's not a big deal. We'll get a second paper towel. I'll just empty that into there. It's a good thing I got extra paper towels, because I know how messy I am. At least it fell into the beaker. Could have been much worse. Could have been much worse. So we're just gonna pop that back in the bag. Then we're gonna strain it again real quick. Give this a quick wipe. got overexcited.
Don't we all? I mean, it is science
is just
after
so
all.
cool. It's just so cool. That aside, put another makeshift filter in there. My allergies are really bad today. Then we're gonna wait.
Meanwhile, I'll sing that hit song from the 90s. I don't want to wait for my strawberries to be straight. Dawson's Creek.
Dawson's Creek. I never watched that. Get this away because this is gonna make it better. Ugh, container. Excellent. Okay, so I don't drop it in again. It dripped. You know what? It's fine. I'll clean it up in a minute. So we have about 50, 60, I think we're gonna get about 70 milliliters out of here. Stop it right there, because I don't want to have space in my medium beaker. There are worse things science could smell like. Okay, so
Oh, I know that.
so we're gonna pour this into our medium beaker. Our 100 ml. Stop right about there. That's 50 milliliters. And then we're going to need a teaspoon of rubbing alcohol. That's this much. Not a whole lot. And just pour it in there. You don't really need to stir it or anything, but you can. And you'll see, if you wait for a little bit, you'll see some white, kind of foamy looking stuff rising to the top. So we're just gonna give it a minute. And then I will show that to the camera so you can see. So you guys probably remember from the density tower that alcohol is lighter than water and also repels water. So when we pour the alcohol in our solution, our DNA solution, the DNA floats out of the water and then just kind of hangs out in the alcohol that's at the top of the beaker. Check that out. Okay, come tell me if this is focused on the ship.
Well, is it what? Oh, hold on. Oh, let me focus on that.
Isn't that cool? So you see that pink layer at the bottom, and then the clear layer at the top, and do you see what's in between it? Kind of a pale pink, fluffy looking layer. That is DNA. That is a big cloud of strands of DNA that we have extracted from this strawberry.
I know what that looks like. It looks like ectoplasm from Ghostbusters.
It kind of does.
Yeah. We should play some music and see if it moves.
Is it focused on me again?
YOLO!
Did it refocus on?
Huh? Oh yeah, you're
It's in
good.
focus. Okay, excellent. Okay, so I'm gonna take my glass rod and swirl it around. There we go. Oh, there's not a whole lot. I wonder what happens if I have more alcohol. Just a little stick. There's not a whole lot of DNA in here, but I am picking a little bit of it up on the end of this stick. Yeah, these little chopsticks. That will help. It does not help. That also doesn't help. This is so frustrating. Anyway, I can try to show it to the camera, but I don't know if you'll be able to see it. Maybe we just need to wait longer. Because I can see it. It's there. I just can't get any of it to stay on the... Maybe like a drop or two to stay on the stick. What do you think, Orlando?
Well, we could wait.
We could wait. Orlando can cut the boring parts out later.
That's like 90% of it. Just kidding! How much longer will this take? Hi, my name is DNA. Wait, what's it doing?
So I can see a cloud of DNA just rose out of that pink layer that was at the top because I added more alcohol and floated to the top of the next layer of alcohol.
Oh no, I hope it doesn't explode.
I wonder if I can pick it up. I think I can pick it up now. There's not much of it, but it's there. I'll try to hold it closer to the...
Oh, let me focus on that.
That is strawberry DNA.
Oh!
The fun thing about strawberries is they are octoploid. So humans are diploid, which means we have 2 chromosomes. I don't know. Strawberries have more chromosomes, so they have more DNA.
It looks like you're gonna make a strawberry Cthulhu. You are a mad scientist.
I am a mad scientist. Yeah, it looks like snot. But anyway, that's DNA.
I hope it doesn't start moving. DNA.
DNA. And you can do this in your kitchen.
Hey,
Have fun.
wait a minute.
What?
What about the
Oh,
variables?
variables?
Yeah, real quick.
You could try different fruits,
Oh, yeah?
different amounts of soap, salt, water, alcohol. Wait different lengths of time, like wait 5 minutes, wait 10 minutes, see what happens. That's about all I can think of. You got any ideas?
Watermelon.
Oh, watermelon. Now I'm hungry.
Watermelon DNA.
Okay, well, I'm gonna go eat the rest of these strawberries. See you later.
Bye.